Well, my name's Eric Padron. I'm an assistant member in the Malignant Hematology Department at the H. Lee Moffitt Cancer Center.
My name is Kathy Smith. I'm an assistant professor at UCSF.
My name is Thorsten Aferlach and I'm coming from the Munich Leukemia Laboratory.
I'm a physician scientist in the leukemia division, and really my role is to spend most of my time performing research in the laboratory and in the clinic so that discoveries that we make, understanding the genetics of myeloid malignancies can be translated to the clinic quickly.
I focus primarily on the care of patients with acute myeloid leukemia, and I do research on FLT3 inhibitors in AML, and primarily focused on mechanisms of response and resistance. I do both clinical research and translational research.
I'm a physician and I treat patients every day, and I also do research projects on morphology as well as on next generation sequencing.
The vision for our research is to serve as the bridge between the large genome studies that have resulted in an exponential gain in our understanding of the genetics of leukemia and the patient at the bedside.
The use of molecular testing for MRD is going to be incredibly important going forward. We're starting to understand more and more in this era of targeted therapy how important these newly discovered molecular lesions are, both in leukemogenesis, predicting response to therapy, and also relapse mechanisms. So it's going to be incredibly important for us to be able to detect these lesions going forward in a very sensitive manner.
My vision for clinical research is that we can put patients on such an assay as the myeloid panel gives us, because I see a lot of unmet needs for research projects right now to investigate patients on molecular markers, on genes of interest.
The Raindance Myeloid Panel offers, or has the potential to offer, several things. I think one, it may reduce the deamination reactions that you see because they are not amplified as it occurs with other amplicon-based sequencing panels. And number two, I think the fact that you can more reliably construct the library so that library quantification and other quality metrics aren't necessarily required in this protocol allows for a more streamlined process.
I think Raindance offers an ability to break down a complex problem, which is tumor heterogeneity, both on the molecular level, which we know is really prevalent in cancer therapy today.
So from my personal perspective and from my personal experience, the Raindance Myeloid Panel really offers us the opportunity to investigate a lot of genes in parallel in a very short turnaround time with a very high sensitivity.